We have Professor Michael Laflamme, who is a senior scientist at the Toronto General Research Institute, and McEwen Chair in Cardiac Regenerative Medicine, at the University Health Network. And we'll talk about novel therapies, again, with the theme of pluripotent stem cells, and he'll talk today on development of regenerative medicine solutions to heart failure.
So I'd like to begin by thanking Mansour and the other organizers for the kind invitation. And I thought for my talk, I would first give you a little bit of an overview about what's been done in the field globally in terms of regenerative medicine and applying it for heart failure. I'm sympathetic with Phyllis and Anthony from this morning. That's a big topic, so I'll give you kind of a very quick overview of what's been done. I thought I'd tell you a little bit about what we've tried historically in this area, and since I'm new to Toronto, I thought I'd tell you about what we're planning to do going forward. So no disclosures. So I thought I'd begin with this slide, and this is, of course, a picture from zebrafish, and it's been alluded to by many other speakers that, of course, the zebrafish and other lower vertebrates are capable of tremendous cardiac regeneration after injury. There's a series of injuries from a landmark paper from Ken Poss back in 2002, where he showed you could snip off as much as 20% of the zebrafish heart, and there was this tremendous regenerative response such that by 2 months later, it was essentially indistinguishable from uninjured controls. Subsequently, they and other investigators have used elegant fate mapping studies to show that this regenerative response involves the proliferation of pre-existing, that is, committed cardiomyocytes, rather than significant involvement by multipotent cardiovascular progenitors. Now, the good news is mammalian hearts are capable of similar regenerative responses, but only very early on. And this is work that Hesham showed in the neonatal mouse heart. Unfortunately, this regenerative capacity in the mouse heart is lost by 7 days after birth. And thereafter, mammalian hearts all repair by scarring. And this, of course, brings us to the problem that will be familiar to all the clinicians in the room, and that is adult human hearts repair by replacement with fibrosis. And this is actually an image that comes from the other half of my day job as a diagnostic cardiovascular pathologist. This is a transverse slice of muscle. Whoops. Is there a pointer here? A transverse slice of heart muscle from an unfortunate individual who died, and we received his heart at autopsy. And you can see they suffered a large anteroseptal infarct, and the infarcted segment, as is typical, has been replaced by this thin, tan, yellow band of scar tissue. And you can appreciate the relative hypokinetic nature of this segment because adjacent to it, we have the formation of this large mural thrombus. And the lower image shows you a typical photomicrograph of what a lesion like this would look like. And that, of course, brings us to the rationale for cardiac regenerative medicine, which is obvious that human hearts do not regenerate like zebrafish hearts. And that brings us to the sobering statistics that you've heard from other speakers that are associated with ischemic heart disease. It's been estimated that up to 1/4 of patients who suffer an MI will go on to develop some level of clinically significant signs or symptoms of heart failure. Numbers for Canada alone, we have about 70,000 MIs each year, and these account for at least a plurality of the 50,000 new diagnoses of heart failure each year. And you've heard about the societal and personal costs of that from other speakers. Now, the good news is medical management for post-MI heart failure has improved tremendously in recent years, but we're still in the situation that the only clinically available means of replacing the lost muscle is to give the patient a whole new organ. And as you all know, in end-stage heart disease, there's an inadequate number of suitable donor hearts to meet demand, and that's what's really driven interest in recent years in novel alternative strategies as a better means to achieve heart regeneration. And again, I don't have time to go into all of the experimental approaches that have been tested in the field. I would divide them, generally speaking, into two broad categories. First, there's a number of investigators that are interested in enhancing those endogenous reparative mechanisms as they occur. And it's probably fair to say that more investigators have been focused on the delivery of exogenous cardiomyogenic cell types. Both of these strategies have their pros and cons. With regard to augmenting endogenous regeneration, the chief limitation there is you're starting off with a very low baseline. And I'd like to underscore a very elegant fate mapping study that was reported by Rich Lee's group a couple years ago. In this study, they combined genetic labels with a method of birth dating cardiomyocytes that involved stable radioisotopes. And in that study, they infarcted these transgenic mice and showed, looking 8 weeks post-MI, that you had only gotten 3% new cardiomyocytes. And they went on to, again, to use these elegant fate mapping approaches to show that that small level of cardiomyocyte renewal was essentially entirely due to cardiomyocyte proliferation with negligible contribution by multipotent cardiovascular progenitors. Again, there's a number of strategies that have been envisioned to try and augment this process. We've heard about this from a number of speakers, including approaches to try and enhance cardiomyocyte proliferation by manipulating cell cycle genes. We heard about follistatin in the preceding talk. There are groups that are interested in trying to activate these resident cardiac progenitor cells, but I think it's fair to say that studies like the one that I alluded to a moment ago from Rich Lee's group are increasingly putting constraints on the functionality, and other studies questioning the reality of some of these putative cardiac progenitor cell populations. And as some of you may have heard, there are other groups that are interested in harnessing what we've learned in recent years about cellular reprogramming, with the idea here being that you might be able to reprogram fibroblasts, which are, of course, a cell type that are very abundant in injured hearts, directly into cardiomyocytes in a novel gene therapy approach. As I said, historically, there's been more activity in the field towards this, with this approach of delivering exogenous myogenic cell types. Here, the limitations are that the typically poor survival and integration of the delivered cells. Certain cell types obviously have their unique safety concerns as well, and we'll talk about one of those in a moment. Again, I don't have time to go through all of the candidate cell types that have been explored in the field. Early proof of concept came with transplantation of primary cardiomyocytes isolated from either neonatal or fetal sources. These studies show that you could get remuscularization of the infarct zone and improvements in contractile function. Historically, a variety of adult stem cell types have been tested. We now are at the situation about 3,000 human patients have received either mesenchymal stem cells or other derivatives that can be isolated from the bone marrow compartment, and there's still a lot of ongoing activity with resident cardiac progenitors. I do think it's fair to say at this point that there's consensus in the field that to the extent these cells mediate beneficial effects, those effects are indirect and are mediated via paracrine mechanisms of action, and that these cells don't significantly repopulate the injured heart with new muscle. And it's that reality that's led our group to focus historically on pluripotent stem cells, because these are really the only stem cell type that's been shown capable of giving rise to large quantities of phenotypically unambiguous cardiomyocytes. And you've heard from other speakers about the two broad categories of pluripotent stem cells that are available to us, embryonic stem cells and induced pluripotent stem cells. And over the years, we and other investigators, and our previous speaker has contributed a lot to our understanding of some of the regulatory factors that drive cardiogenesis. We've figured out ways to guide these cells that could give rise to any of the differentiated cell types of the postnatal organism to become increasingly restricted multipotent cardiovascular progenitors. All of the parenchymal cell types that would be required to repopulate injured hearts, including cardiomyocytes. And works I'll highlight in a few minutes, we now have the capability to go one step further and get relatively purified populations of ventricular, atrial, and nodal cells that you could think about using for downstream clinical applications. This is a movie to show you what these human hESC-derived cardiomyocytes look like in the dish. One of the fun things about them is that they do what cardiomyocytes are supposed to do. They show contractile activity. Again, this spontaneous contractile activity is actually a marker of immaturity, but it still gives you a nice quick readout that you've in fact got a preparation with a high percentage of cardiomyocytes. And work that we commenced with my former colleagues at the University of Washington, including Chuck Murray, over a decade ago, we showed we could take the relatively crude human hESC-derived cardiomyocyte populations that we could isolate at that point in time and transplant them into rodent models of myocardial infarction, first working in athymic rats. Directly inject those into the infarcted segment and get partial remuscularization of the infarct zone. This was actually the median example from this study, in which we saw these islands of human myocardium growing out in the scar tissue. And we knew they were cardiomyocytes because they were positive for cardiac markers like beta-myosin heavy chain here depicted in red. And we always confirmed their human donor origin by in situ hybridization with a human-specific DNA probe. And these cells also expressed other expected cardiac markers like cardiac troponin I. In this early study, we also provided the first evidence for functional efficacy with the transplantation of these cells. This is data that was obtained by echocardiography looking at fractional shortening. It's a busy graph. I'll draw your attention just to the red bars. What I'm showing you here is fractional shortening in the cardiomyocyte recipients going from 48 hours pre-transplantation to 4 weeks post-transplantation. And we saw complete preservation of fractional shortening in those recipients, an effect that was not seen in any of the 3 control groups. Now, importantly, we did not see restoration of function back up to levels seen in uninjured controls. In this study, we also used cardiac MRI, which is, of course, nice because it can give you an assessment of regional wall motion. And again, we saw an improvement in wall thickening that was unique to the cardiomyocyte recipients, not seen in the controls. But we did not see restoration of wall thickening back up to the uninfarcted segment or in uninjured controls. I want to highlight a more recent study that was actually reported late last year by Chuck Murray's group, where for the first time they did a head-to-head study, again working in the rat infarct model, in which they transplanted human hESC-derived cardiomyocytes, multipotent cardiovascular progenitors from these same ES cells, and bone marrow-derived mononuclear cells, which is the cell type that's been most extensively used in human patients. And I think going forward in the cell therapy field, there's going to be more of a role for these kind of direct empirical head-to-head comparisons. And interestingly, what they found is that there were no improvements in left ventricular dimension or fractional shortening in the bone marrow cell recipients. We saw a similar improvement, however, in those parameters in both of the cardiomyocyte and the multipotent cardiovascular progenitor recipients. Interestingly, although these multipotent progenitors were capable in the dish of giving rise to endothelial cells and other vascular structures, they did not significantly contribute to those in vivo. So I don't think at this point there's good evidence that those cells outperform cardiomyocytes, but that will require further study. So I told you what's been done in the rat model historically with these cells. A few years ago, my laboratory invested the effort to develop the guinea pig as what we thought would be a more relevant small animal model for transplantation studies with these human cardiomyocytes. And our primary considerations here were electrophysiologic. The guinea pig is a much better match to humans than are mice and rats, and their sinus rate is a lot closer to humans. So the sinus rate of a mouse is about 600 beats per minute, rats about 450. Guinea pigs are still a lot faster than you and I, but they have a sinus rate of about 200 beats per minute or so, which is just comfortably below the maximal rate at which we had found in previous studies these cells would respond in vitro. In terms of histologic outcomes, we saw very similar results to what we'd seen previously in the rat, again with partial remuscularization of the injury zone and improvements in parameters like fractional shortening. But our main question in this study was whether the transplantation of these cells led to the formation of graft myocardium that would become electrically integrated and fire in synchrony with host tissues. And we knew that these cells had the subcellular machinery necessary for that electromechanical integration. And we could tell that histologically. We could identify points of contact between host and graft cardiomyocytes, and in between them we could see immunoreactivity for the gap junction protein connexin 43. We also saw elements of the mechanical junctions between these cells, including end-cadherin. But to more directly address whether these cells were functionally coupled, we actually engineered human ES and later iPS cells that express a genetically encoded calcium sensor molecule. This is the fluorescent protein GCAMP3. And you could think of GCAMP3 as combining green fluorescent protein with the calcium binding moieties of calmodulin. And so in the presence of elevated calcium, this molecule undergoes a conformational change and an increase in fluorescence. And the long and the short of it is cardiomyocytes expressing this reporter show robust fluorescent transients with each contractile cycle. This is a cartoon to show you sort of the schematic for these studies that we later did in the guinea pig. We would typically injure the animal, intervene with a repeat thoracotomy and transplantation of 100 million of these GCAMP-positive cardiomyocytes, either doing so 10 days post-injury to kind of mimic a subacute injury model, or 28 days later, at which point you had established heart failure in this model and well-established scar. And then we would follow these hearts and do ex vivo imaging studies either 14 or 28 days post-transplantation. And of course, here the readout is relatively straightforward. If we see a flash, we know the graft is active, and then we can correlate those flashes with the host electrocardiogram. And what I'm showing you here is a montage of images that required in normal, that is, uninjured guinea pig hearts. And I think you can see it looks like all regions of the graft are flashing, and they seem to be flashing in nice one-to-one synchrony with the QRS complexes of the host electrocardiogram. Now, you may have a concern here about motion artifacts. The heart's obviously moving in and out of the plane of focus. And so to address that, we can take these hearts out of the chest, keep them alive ex vivo in a Langendorf apparatus, and under those conditions, we can perfuse the heart with drugs like blebistatin or BDM that cause mechanical arrest but persistence of electrical activity, and we still saw nice one-to-one coupling under those conditions. And we really like that preparation. It allows us to be a lot more quantitative. We can grab graft regions of interest, plot their fluorescent transients over time, and line them up against the electrocardiogram, and actually ask what fraction of the graft is coupled. And we've now done this. This slide is actually a little out of date. We're approaching 24 uninjured guinea pig recipients, and always found 100% coupling over 100% of the visible graft footprint. We could pace these hearts up to about 5 hertz or so and still get nice one-to-one capture. Now, it's hard to prove a negative, but in my lab, we've looked at this pretty carefully, repeating these experiments in the rat model, and never seen convincing coupling in what I would consider a normal rat heart. If you slow a rat down to 60 beats per minute, and we could argue about whether it's still alive under those conditions, you can occasionally see coupling, but it's usually the graft driving the rat rather than the other way around. And so to us, this really validated the investment we've made to develop the guinea pig as a better small animal model. So the data I just showed you came from transplantation studies in normal, that is, uninjured guinea pig hearts. And that's a very different context than would apply in the injured heart, where you're going to have all of this intervening fibrosis that's going to act as a barrier to integration. And that's indeed what we found to be the case. So here I'm showing you one of our successes. So just to orient you, the injury zone, the border zone's running right along here. And we have 2 graft regions. One's in the border zone and one's protruding out into the scar. The field of view is about 2 centimeters here. And you can see in this case, both graft regions are nicely one-to-one coupled to the host electrocardiogram. Unfortunately, that wasn't always the case. So we had other hearts like the one I'm showing you here, where we had, in this case, if you watch carefully, the red graft region is nicely one-to-one coupled to the host electrocardiogram, but the blue one is not. It's essentially marching to its own drummer. So in a heart like this, we don't just have a problem of host graft integration that we need to improve, but also graft-graft integration. And this is a major focus right now in the laboratory. So in recent work that I don't have time to go into the details, we've been developing other tools by which we can, in detail, characterize the electrical behavior of both host and graft tissue in engrafted hearts. And in the course of this work, we've identified some novel fluorescent voltage-sensitive dyes that we can perfuse into these hearts and use them to track separately what's going on in graft and host. Here I'm going to show you in a minute a video that's actually reporting what's going on in both tissues simultaneously. And just to orient you, in the video I'm going to show you in a second, the injury zone is represented by this dotted area right here. And this map that you see sort of looking like Antarctica in the middle is your graft tissue, which is occupying, in this case, most of the scar tissue. And in this video I'm going to show you in a moment, there's actually a pacing electrode on the left ventricular apex here. And you're actually going to see activation going from the right-hand side of the field up towards the left. And I'll show you the simultaneously acquired electrocardiogram. And so you're going to see normal electrical systole here, going again from right to left. You're going to see another electrical systole in a moment. And then I want to draw your attention to what looks for all the world like a premature ventricular contraction here. I want you to see there's a very different pattern of activation in that instance. So actually here you're going to see the activations occurring in the middle of the field, which is right smack in the middle of the graft tissue. So this is an example of the graft acting as an ectopic pacemaker. Just to illustrate how different these individual beats were, here I'm showing you representative activation maps for the typical host pace beats and the graft pace beats. And we've seen in this model multiple instances of where these grafts can act as an ectopic pacemaker. Now, this wasn't a surprise to us. In these studies, we're using relatively impure preparations of cardiomyocytes. And we know from other studies that these include anywhere up to about 1/3 nodal pacemaker cells. And so it's pretty easy to imagine those could be partially responsible for this process. So in the last few minutes, I'd like to tell you about some of our past and future work in large animal models with these cells. And this is a study that we completed a couple of years ago with Chuck Murray's group, where for the first time we wanted to transplant these cells in a large animal model. And so we settled on the non-human primate, and in this model delivered 1 billion human ES-derived cardiomyocytes, again, directly injecting it into the infarct zone. And this, again, is the median example I'm showing you here. The dotted line is actually the perimeter of the infarct scar here. And you can see that we've remuscularized, and this was typical, about 1/2 of the infarct footprint with, in this case, GFP-positive human myocardium. And so this is actually a greater degree of remuscularization than we had obtained historically in small animal models. So we were very excited to see this. Another encouraging result from the study is we saw evidence for structural maturation of these grafts over time. So we can get these cells much more mature in vivo at this point than we're capable in the dish. And just to illustrate that, here I'm showing you immunostaining for alpha-actinin, looking at a representative animal at 2 weeks post-transplantation. You can see they have sarcomeres, but those sarcomeres are sort of infrequent and haphazardly organized. By contrast, in an animal after about 3 months in vivo, these show much more sarcomeric organization, and if you actually look at cardiomyocyte dimensions, they're essentially indistinguishable from the adjacent host cardiomyocytes. And so one of the things we're very interested in asking is whether this structural maturation is accompanied by functional maturation. That's an unknown right now. We also in this study used that GCamp reporter that I described to you before to show that in this model, if anything, we got better electrical mechanical integration than we had seen historically in small animal models, probably in part because we were just simply getting better, larger grafts. So this is the good news. The one bad news that emerged from these studies, and this again wasn't a complete surprise to us, is all of the recipient animals showed transient bouts of non-lethal ventricular tachyarrhythmias. And the joke here, but it's true, the animals were tolerating this quite fine. The monkey sitting there eating a banana, we're the ones that were looking at the rhythm strip and feeling the chest palpitations. But obviously this isn't something that would be tolerable in a human patient that's suffered an infarct. And these animals showed a variety of abnormal rhythms, ranging from accelerated idioventricular rhythms to bouts of sustained and non-sustained ventricular tachycardia. There's an interesting time course to these events. They seem to peak about 2 weeks post-transplantation, dissipated substantially by 3 weeks, and very small ends here, but we didn't find any arrhythmias looking 4 weeks and beyond. And so one possibility is that this structural maturation was accompanied by electrophysiologic maturation, which may account for their greater electrical stability over time, but this is something that will have to be investigated further. So to summarize what I've told you so far in terms of preclinical studies with these human hESC-derived cardiomyocytes, with studies ranging from transplantation into 300-gram rats with a sinus rate of over 400 beats per minute to up to 10-kilogram primates that have a sinus rate, again, still faster than you or I, but anywhere upwards of about 130 beats per minute. So in all of these animal models, we've shown that we can get an engraftment. And in fact, the engraftment is more robust in the large animal model. And when we've delivered cells of sufficient purity, we've never seen tumors. In fact, if you look 2 weeks and beyond, you have to look really hard to find even a single persistent non-cardiac derivative. We've shown improvements in contractile function in the small animal models. That is not something that's been shown by us or, to my knowledge, by anybody in a large animal model. So that's a major goal for us in the field. And we've demonstrated that there can be host graft electrical coupling once you're talking guinea pig and larger. And perhaps as a consequence of that electrical coupling, we've seen these non-lethal ventricular tachyarrhythmias in the primate model. Now, we learned a tremendous amount from all of these models, in particular from this work on the non-human primate. But I would submit that the non-human primate still has some significant limitations for preclinical studies. These are obviously much smaller than humans, and their sinus rate is still faster than humans, which is a concern given this electrophysiologic phenomenon. And so in work that we're just rolling up our sleeves and getting underway here in Toronto is we're doing transplantation studies with these cells into pigs. Obviously, pigs can be 30 kilograms and larger and have a sinus rhythm that's much closer to you or I. So what are the remaining challenges in the questions we're trying to answer in this model? Well, in my mind, the biggest safety issue that we need to tackle is we need to figure out ways to stabilize the electrical behavior of these engrafted hearts. And we think the pig will be a better model to address those questions. We still need to demonstrate efficacy in a large animal model to justify moving forward, I would say. We need to establish scalable and economically viable means to produce cardiomyocytes, and ultimately this has got to be done at clinical GMP grade. We need to identify an optimal strategy to try and prevent graft cell immune rejection. So I'm very pleased to report that about 3 or 4 months ago, we were fortunate to get a grant from the Ontario Institute of Regenerative Medicine to try and answer some of these questions in the infarcted pig heart. And our long-term goal in this study is to try and obtain the preclinical data that we would need for an eventual clinical trial application to test not just human hESC-derived cardiomyocytes, but specifically human hESC-derived ventricular cardiomyocytes and post-MI heart failure. And the themes here are what we think is a better animal model and ultimately better cells. And the key enabling technologies is Gordon Keller's lab has developed greatly improved protocols for guiding the differentiation of human embryonic stem cells into ventricular derivatives. And in the dish, those cells are nearly electrically quiescent. They show spontaneous beating rates that are actually substantially below the junctional escape rhythms of the human heart. So we think that those will be more stable and show less predilection for ectopy. And also in work that was done by Peter Zanstra and his colleagues at CCRM, they've really worked out large-scale cardiomyocyte manufacturing. And in recent months, we've taken this one step further and actually just generated a batch of over 3 billion cardiomyocytes from a single 3-liter stir tank reactor. So we can generate the quantities of cells that would be necessary for large animal studies and going forward for humans. And we're deploying this in a GMP pedigree human ES cell line. So our goals in the study are to try to optimize, again, the scaled and ultimately GMP-grade production of these ventricular myocytes. We want to test, this will be the first experiment we'll actually do on the pig, is to test the hypothesis that the transplantation of these ventricular cells will actually give us better outcomes in terms of arrhythmogenesis, comparing them to the historical mixture of cardiac subtypes that we've transplanted in the past. And ultimately, we need to determine whether their transplantation has any beneficial effects on contractile function and what will be a pivotal efficacy study. And I'm pleased to report we actually did our first transplant of cardiomyocytes actually this past Monday and have on the calendar 1 animal per week for the next few weeks. So to conclude, I'd like to thank the many folks that contribute to these studies. Most of the work I told you was actually done at my previous institution at University of Washington, and many of these were done in close collaboration with Chuck Murray's group there. And I'd also like to thank, I didn't really show you about some of our more recent work, but we're really excited to get our sleeves rolled up and working with my new colleagues here in Toronto. And with that, if there's time, well, I guess we're doing questions at the end. Thank you for your attention.